AS Level Biology 9700, topic cell structure. This is the last video on this topic, and today's subtopics are, first of all, we will discuss the prokaryotic cells. So if we see the structure of the prokaryotic cell, you can see that a flagellum is there. Plasmid is the circular DNA which is present in, and you can see this DNA or nucleolide, they are present in the center without any membrane. So it means membrane is absent. And it is present in the center. Ribosomes are there, cytosols are there. You can see the plasma membrane is also there. Cell wall is there, capsule, a protective covering. Capsule is protective covering. So flagellum is for movement. So in this way, these are the different parts of a prokaryotic cell, because the prokaryotic cell is that cell which don't have a membrane bound nucleus, or the organelles are without that membrane. So a proper nucleus is not present in prokaryotic cells. So the prokaryotic cells lack a nucleus and most internal structures of eukaryotic cells. Different structures, organelles properly, which are present in eukaryotic cells, they are not present in prokaryotic cells. So they are different. So no membrane bound organelles. All organelles which have membranes around them, they are not present. Only the ribosomes have that. So DNA contained in one circular chromosome, which is called as plasmid. So circular DNA is there, which is called as plasmid. So cytoplasm is also present. Cell wall is made up of peptidoglycan. So this is another type of uh, carbohydrate. So cell membrane inside cell wall, cell membrane is also there. Some have flagella for its movement. So flagella is locomotive organ, and with the help of which these organisms can move from one place to the other. So smaller and less complex than, so they are not complicated, complex, just like the eukaryotic cells. And we can give the example of bacteria and blue-green algae. So you can see our bacteria is the best example for when we talk about the prokaryotic cells, because they don't have proper membrane bound nucleus. And you can see the DNA is directly embedded into the cytoplasm. It is present in the center, but there is no nuclear membrane around it, due to which we can't say this is a proper nucleus. So proper nucleus is not in prokaryotic cells, only DNA is there. And beside this, another very important plasmid is present, which is circular DNA, which is very important. So it is also helpful in genetic engineering. We can see that this plasmid has very importance. So prokaryotic cells are very important in this way if we talk about them. So they are involved in different functions. So if we compare the two different types of prokaryotic cells, then we can see that some of them are without flagella, and some of them are with flagella and other uh, parts of the cell. So cell wall is, so we will discuss the structures which are always present in all types of prokaryotes. They are present. The cell wall is always present, and which is made up of peptidoglycan. Cell surface membrane is present, but the different organelles or the nucleus are without this cell membrane. Cytoplasm will always be there. Circular DNA or plasmid will always be there. Ribosomes will always be there. So these are the, you can say, the structure which will always be present in a prokaryotic cells. But in some of the prokaryotic cells, some additional structures are also present. One of them is flagellum, which is locomotive organ. So with the help of which the uh, cells can move from one place to the other. So capsule, which is protective covering. So this capsule is a protective covering. From adverse environment, this protective covering or for protection, this protective covering will be present in some types of uh, prokaryotes. And then you can see the infolding of cell surface membrane may form a photosynthetic membrane or carry out nitrogen fixation. So this is very important function, that is which can be present in some of the prokaryotes. Plasmids, small circles of DNA, several may be present. So plasmids are present in all, but the plasmids can be present in very small. So pili for attachment to other cells or surface involved in sexual reproduction. So pili, you can say, with the help of which they can attach themselves, themselves with some surfaces. And already told you about, that is the plasmid. Plasmids are present in bacteria. So I would like to write over here the example. They have plasmids, and due to which the bacteria has very importance in biology. Now the second type of cells which we are going to discuss are the eukaryotic cells. So eukaryotic cells have proper membrane bound nucleus. A proper nucleus is there. So membrane bound nucleus is there, and the other organelles such as ribosomes, mitochondria, ribosomes, etc. All organelles which have double membrane around them, all they will be are categorized into the eukaryotes. So you can see the flexible boundary of the cell. Cellulose cell wall will be there in case of if there is a plant cell. Separates the cell from its surrounding cell membrane is there. Plasma membrane, endoplasmic reticulum, nucleus is there, ribosome is there, Golgi bodies are there, mitochondria, lysosomes. All these are the different organelles which are present in a eukaryotic cell. So this is the proper cell which you can say we can compare the plant cell, animal cell. Already in the start of this topic, I had discussed all the structure of an animal and plant cell. So if we compare the eukaryotic cell with the prokaryotes, then um, no membrane bound nucleus. No membrane bound nucleus in prokaryotes, but whereas the membrane bound nucleus is present in eukaryotes, cell walls made of peptidoglycan, as for example, the gram positive, gram negative bacteria. So they peptidoglycan, whereas the cell wall is made up of cellulose or chitin in fungi. So the different types, you can say type cell walls are there because the presence of different materials in the formation of cell wall. So no membrane bound organelles are there, as for example, mitochondria, lysosomes, ribosomes, so all, and chloroplast as well. So they are the double membrane structures. Have pili and fimbriae for adhesion and flagella for propulsion or for the movement. So whereas in eukaryotes, cilia or flagella are present. Cilia or flagella are present for movement, for as a locomotive organ, for locomotion, you can say that, and mucilaginous capsule, protective capsule, which is only present in some prokaryotes, not in all prokaryotes, but in case of eukaryotes, there is no need for this mucilaginous capsule. So there are different internal structures present. You can see this. The cell size ranges from 0.5 micrometer to 100 micrometer. So very smaller in size, whereas the eukaryote cell size ranges from 10 to 150 micrometer. So they are larger. Eukaryotes are larger than prokaryotes. And the next subtopic is virus, and this is the last topic of this uh, cell structure. So viruses, virus mean poison. So virus vary in their structure. Virus, the microorganisms which cause different types of diseases, not only in human beings, but also in animals and plants. So the different types of viruses are involved in the spreading of these different types of diseases. So a virus particle consists of DNA and RNA, a protective protein coat called a capsid. So the outer protein cover is there. Inside the protein cover, there is DNA or RNA. You know, DNA is deoxyribonucleic acid, deoxyribonucleic acid, or RNA stands for ribonucleic acid. DNA is double strand structure, and whereas ribonucleic acid is single strand structure. So the capsid is made from the proteins that are encoded by viral genes within their genome. The shape of the capsid serve as one base for classification of viruses. So it means the shape of the capsid will be different in different types of viruses. So viruses can replicate only by infecting a host cells. They can be considered between the living and non-living, because when they are present inside the host, they can replicate. When they are outside the host, they are just like the dead cell. So they cannot reproduce on their own. Viruses are not cells. They are not cells. Viruses are not considered as cells. They are strand of genetic materials with a protective protein called a capsid. So they are, you can say, not a proper cell. They only made up of protein and protein covering or coat, and inner core is made up of DNA or RNA. So it is not a proper cell. We will not say the virus is a cell. So they infect a wide variety of organisms, so they can cause different types of diseases in both eukaryotes and prokaryotes, in animals, plants, and human beings as well. So the different diseases which they can cause are the cancer. They can cause the you can say the different AIDS and other diseases as well. So here you can see one type of virus, and you can see the capsid, which is it's made up of protein, different types of proteins, and you can see the receptor proteins are also present on the capsid surface, and DNA, RNA, but this is the single strand, so it is the RNA. So in some of the viruses, there is double strand structure, which is DNA. So viruses cause different diseases in human beings, animals, and plants. So different types of viruses are there, but they are not considered as cells. So students, it was all about this topic one. We have completed it in 6 videos, but videos are very short. So because it is very time taking, and these sometimes students don't watch the complete videos. So this is the reason I have made very small videos. So it will be very easy for you to learn all these topics or to understand all these topics. If you want anything, if you you can message me, and in this way, I will be able to reply you. Uh, One more thing, please don't forget to like, share, and subscribe my channel. And thanks for watching.